This is a tea tree in India after an insect attack.
This is a wheat field in China after an insect attack.
This is a cornfield in the United States after an insect attack.
This is a rice field in Taiwan after an insect attack.
This is a coffee tree in Uganda after an insect attack.
This is a grape tree in Spain after an insect attack.
This is a soybean field in Argentina after an insect attack.
Harmful insects are causing great damage to our lives.
Each year, agricultural loss caused by insect damage alone is $470 billion.
20% of the annual crop production is destroyed by harmful insects.
If some of this food can be saved from insect damage, it can be used to feed the more than 3 billion people who are starving around the world every single day.
Insect damage has always been a problem, and yes, continue to be a problem, even with the advance in technology nowadays.
This problem has to be solved, and people have never stopped trying.
However, our current solution is pesticide. 3 billion tons of pesticides is being applied this year on crops.
Pesticides can cause severe health problems such as birth defect, nerve damage, and cancer. It is also greatly damaging our environment in all its different forms of devastation. We are the 2014 NCTU Formosa iGEM team.
We need a change.
Good morning, ladies and gentlemen. We are Team NCTU Formosa, and today we are here to give you a briefing on our project this year, Operation Debug. I am Supervisory Special Agent Rachel Lee.
And I am Supervisory Special Agent Nick Shaw.
We are from the Harmful Insect Elimination Force, and our headquarters is based in National Jiao Tong University, Hsinchu, Taiwan. As our name suggests, our task force is dedicated to the solving of insect damage.
Hundreds of thousands of hectares of production is wasted around the world every year.
Currently, our solution is pesticides, yet we know it is a very lousy solution.
$9.6 billion are lost on damage of pesticides annually.
Our goal is to resolve insect damage without the terrible side effects of pesticides. Ooh, boss is calling.
Oh, boss, I'm not ready yet. Oh my God.
Good morning, agents.
Good morning,
Morning, please.
Status update.
In a relationship.
No, you're single, come on.
Agents, status update on Operation Debug.
Yes, sir. As we know, in nature, opposites attract. So we are using this to our advantage. We will manipulate a female moth and allow it to attract more of the same species of male moth. At this stage of operation, we have already acquired the target.
In this operation, we are mainly targeting Oriental default moth. Its main area of activity is tropical islands, and a major part of the damage is done in Taiwan.
This insect is highly dangerous, and it is a serious threat to our nation's security. It feeds on over 40 families of plants, which includes basically almost everything.
It is a danger to every crop.
And we are hunting it down.
So, what's your plan?
We are planning to have a hunt.
Me?
Oh, no, I mean, I was planning to have a honeymoon in Paris.
Use that on your personal leave this year. I'm the one who's going on a honeymoon. So, what's your plan?
Okay, yes, sir. And the honeymoon is...
Nick makes the tactical plan of operation. Sir, I apologize for my partner's behavior. Currently, as we know, the most commonly used method to attract and capture insects is the pheromone trap. So originally, we know that the pheromones are the moth's hormones. and it is originally used for mating and certain other purposes. What's so special about it is that it can specifically attract insects. So we wanted to biologically synthesize pheromones to replace the currently in use chemically synthesized ones. However, we found the biosynthetic pathway of pheromones way too complicated for E. coli to execute. So this mission plan is practically impossible and is thus aborted. We started looking for a plan B, and we decided on infiltration. We are sending in an undercover agent, PBAN, whose full name is Pheromone Biosynthesis Activating Neuropeptide. Agent PBAN would turn our target insects against themselves, prompt them to start making pheromones for us, and in turn, attract more of the very same kind of insects for us. So, how exactly is this going to work? Well, here we have the pheromone gland of our target insect. And once the PBAN binds with the receptor on the gland, biosynthesis will be activated and pheromones will be emitted. So forget this complicated reaction, because we can let the insects do the job for us. I mean, after all, they are naturally designed to do so, right? So now I would like to give more information as to why we chose agent PBAN for this operation. A lot of it has to do with its identity. It is a simple short peptide. As a peptide, it means that it can be easily made by E. coli through expression. The fact that it is smaller than 150 base pairs is another plus because it wouldn't place any stress on the E. coli for expressing it. As a peptide, yet another advantage is that it wouldn't be easily degraded because it's already in a very, very simple form. Now, the most important part is that it is specific. PBAN has the same species specificity as pheromones. One kind of PBAN can only react with one kind of insect who will in turn release its own pheromones and attract more of the very same kind of insect. So if we allow 3 different kinds of insects to come in contact with the same kind of PBAN, what will happen is only the corresponding kind will react. In this case, we are using the moth's PBAN. So only the moth will react with the moth's PBAN, release moth pheromones, and attract more moths. Now, I would like to talk about our biobrick assembly. As you can see, it is fairly simple. We use a constitutive promoter and combine it to our ribosome binding site and the DNA sequence of our PBAN. We named this PBAN biobrick PBAN SL, taking the first letters of the Latin name of Oriental leafworm moth. So far, what do you think, boss?
Very well. So, where are you in your plan now?
Okay, so to confirm that E. coli can actually make PBAN, first of all, we did a PCR test, putting the forward primer before the promoter and the reverse primer after the PBAN sequence. The theoretical length should be around 600 base pairs. And as you can see, the result is a clear single band right between 750 base pairs and 500 base pairs. So this informs us of the first step of our success. Now, we wanted to really see the expression of our peptide, so we ran an SDS page del. And again, the result is a clear band compared to the loading control right at 3.7 kilodaltons, the correct length of our peptide. So this means that our PBAN is successfully expressed. Then we constructed another biobrick assembly tagging a BFP gene after the PBAN as a reporter gene. The result is that all the E. coli gives off blue fluorescence, meaning again that PBAN is successfully made, but this is not the only purpose of tagging the BFP gene. We use the fluorescence intensity to find the best time point to gain the maximum yield of our PBAN. So, now that we know that our E. coli can indeed make PBAN, what we want to do is take this PBAN, make pheromones, and let the actual attraction happen. From this diagram, we can see that PBAN binds with a G protein coupled receptor that once activated will change the calcium ion concentration, initiate the calcium pump, and then activate the entire biosynthetic reaction of pheromones. So, our PBAN is already made inside the E. coli. How exactly are we going to use this PBAN? To overcome the cell membrane barrier, we did something very simple. First of all, we pour out the liquid broth, and then we added some buffer, and then we mash this entire solution. breaking the cells, letting the P-band to come out of the cells and into the solution. And then we add more water or less water, depends on what's your desired concentration, and then we take it to autoclave. Then we obtain an E. coli-free, biologically safe P-band solution. Now, we soak cotton into this solution, and then we sprinkle sugar on the cotton. So once our target insect comes to eat the sugar on the cotton, they will directly come in contact with the P-band on the cotton and release pheromones, of course. Now, you can see here on the left, we have a female moth ingesting a sugar-only solution with no P-band inside, and on the right, we have the same kind of female moth under the same kind of conditions ingesting the P-band solution we made. You can clearly see on the left there is no change in the moth's behavior before or after the ingestion. But on the right, this female moth is frantically flapping its wings. This is a very, very important milestone of our project. This means that our P-band works. So from this point on, this female moth is officially sexually stimulated, and it is giving off pheromones to attract more of its male counterparts.
Wonderful! And what weapon are you going to use for this operation?
Well, a large caliber handgun wouldn't do it, a Glock would have too much recoil for accurate aim, a SIG, a Beretta, I don't think any of our currently existing weapons would do the job. What are we going to do, Mick?
Okay, so we have to design our own weapon. Please say hi to my baby. Hi! Hi. Just as we previously mentioned, we hope to not only establish a new solution to insect damage currently happening all over the world, but also apply that solution to agriculture and even everyday life. For example, your backyard. You can see on the side that our device is like a pyramid. We choose acrylic panels as our material in order to ensure it is strengthened enough to support the device and not easily destroyed by pests. Now, let me introduce the detailed structure of our device for you. It can be divided into several parts. First, the base. At the center, we design a red string-like structure to hold our plate, and another red square structure for the inner shell to be clicked on. Next, the outer shell. It is formed by 4 equilateral triangles. The reason why we choose a pyramid to be our shape is that it facilitates draining of water and snow and prevent pounding. This allows our device to function normally in any weather condition. We also design a pyramid with its top part traveled as an inner shell. We continue to use a pyramid shape not simply for overall design and consistency, but also create a smoothly inclining duct for the target harmful insect to easily enter our device and unable to escape. We placed LEDs on the inner surface of the inner shell. They are powered by a battery will be positioned here. This is not only for making our device look cool at night, but it plays a crucial role of attracting the first target insect into our device and make pheromone for us. So now, it's time to tell you how we use our device. To make insect helpers, first of all, we have to make insects come to us. To do so, we will use light to attract them into our device. Once inside the device, they will be led to our P-band by the sugar we coated on our P-band salt cotton. Until this point, there is no specificity in the attraction. However, once the target insect inside enters our device and ingests its own P-band, it will release pheromone. Then more of the target insect will be attracted into our device. So now our target harmful insect has already released its own pheromone, so even if we turn off light, we can still attract them into our device. And we did an experiment using our device to see the attraction effect of blue light and P-band. Here we have a result. You can clearly see that blue light alone can attract much fewer moths than the combination of blue light and P-band with our system uses. So now, we have a video of our device working its magic. Take a look.
In this experiment, we used a chamber that simulates the natural environment. We placed vegetable leaves and flowers, which are both appealing food sources to the moths. There are also around 150 male moths placed inside the chamber for experimental purposes.
Now, we place a female moth into the speaker and it will ingest P-band. After everything is set, we place a pyramid into a chamber to begin our experiment.
Initially, you can see that all of the male moths were just wandering around and minding their own business, not really doing anything in the chamber.
But after our system takes effect, to our excitement, they were all attracted into our device.
After 11 hours, we captured dozens of target insects into our device, and none of the unrelated species were affected at all. With our new method, we are trying to start an agricultural revolution. The concept is eco-friendly, easy to use, low cost, and fashionable.
Our device is part of this revolution.
Not only does it effectively attract and capture harmful insects, but it will become a must-have garden decoration in the future. It will light up your garden, or your farm, or wherever you choose to hang your device, like a beacon in the night.
This is our device, and it is magically beautiful. Debugging style,
we are your best choice.
Help farmers find the best condition to use our device. We did modeling on how little moths will behave under different lights and temperatures. For example, if it is 22 degrees today, farmers can look at our modeling result and choose blue light, which will give them the best result of insect attraction. However, you can see from the result, the blue light can attract much more moths than any other light in all temperature conditions. So we use blue light in our device.
Now that our biggest threat is eliminated, we are taking a step back to continue to help the world to fight the rest of its evils. You can see that all of these insects are also currently causing great damage to our entire world. So we constructed biobricks with their P-bands as well. With a little bit of effort, all of their problems can be solved by our system and our P-bands. Now, I would like to proudly present to you the results for all of these P-band biobricks. First of all, the PCR test confirmed that we successfully cloned all of the P-band genes onto their separate backbones. And then the SDS page gel results showed that there are clear bands at the separately correct positions for all of the P-bands. So this means that all of the P-bands are indeed successfully expressed. The BFP tagging result showed that all strains of E. coli give off blue fluorescence. So again, this means that all of the P-band are successfully expressed, and we can use the fluorescence intensity to find the different yields at different time points.
As fine as your work is, our agents alone cannot accomplish this task. A hero can be anyone. We just have to find them. And this is done through the practice amongst humans.
Yes, sir. Through our human practice, we not only took our project to the industry, we also educate people, be farmers or students, of the importance of protecting our environment. Our human practice can be separated into 3 criteria. First, also the most significant factor of our operation is the people who support us. We cooperate with some organic farms to take our operation out of the lab and into reality. We not only call our target insect at this farm to complete our testing, we also provide them with our device so that they can use it and give us some feedback. Second, we're the mini meetups with our iGEM team. This summer, I'll held the 2014 iGEM conference, inviting 22 teams and over 150 people in Taiwan and China to attend. In this meetup, we shared our project with each other and exchanged thoughts and ideas in hopes of perfecting our project and also getting to know more iGEMers around the world. Third, we promote iGEM with a passion. We held a biocamp this summer to introduce iGEM to senior high school students. In addition, we talked to farmers at the Green Farmers Market Fair about our project. With their everyday farming experience, they gave us a lot of useful advice. They are amazed by the concept of iGEM as well, and were very excited to cooperate and help iGEM teams bring better lives to people.
We've talked about so many things, but what exactly have we achieved this year? First and foremost, we built biobricks, of course. We converted 9 P-bands into standard biobrick parts, and for each of the P-bands, we built a composite part with the promoter and the RBS, and another composite part with the BFP tagging function. So in total, we constructed 27 new biobricks this year. Secondly, we successfully resolved insect damage using our new system and crafted a new device allowing the application of this system. thirdly, we not just stay in the lab and say, ooh, everything works perfectly on paper. We really walked out of the lab into the industry and took our system into the field, into the everyday farming environment. Fourth, we held the iGEM conference and inviting 22 teams from China and Taiwan and over 150 people from many different countries. We also met up with many different other iGEM teams, and all of it has been a memorable journey. Last, but definitely not least, we provided modeling assistance to TCU Taiwan iGEM team. Now, I would like to give thanks to all of our instructors and alumni sponsors for their support, and also every single member of this team. Thank you for making NCTU Formosa the team we are today. This is the end of our presentation.
Thank you for your attention.
Thank you.
Thanks, guys. Just a quick question. Did the rest of your team shows up?
Yeah, sorry.
I'll let you
Give us a sec.
What kind of ethics approvals did you need to use the moths in your experiment?
The moths?
Yeah.
Mm.
Or if you didn't require approvals in your institution, what kind of considerations did you give to it?
Oh, okay. So the moths we use are caught around our campus and also in the organic farms we cooperated with. And frankly, we haven't really, we didn't have much regulation considering this ethical problem. So we didn't really have any official approval from anyone. But we did treat the moths right. We give them enough nutrients and we keep them from working too hard. Like we would give them resting time for each of the experiments and yeah, this. Thank you for your question.
I had a question about when you were trying to figure out what the best time was to make your cell lysate. That was by looking at the BFP fluorescence.
Yes.
And do you think that very high BFP fluorescence will necessarily correlate with the most of your peptide output?
We believe so, of course, but we actually did modeling on how a vector with only the BFP gene would behave and the BFP fluorescence of our composite part to see if the results are meaningful and would have any significance. Yeah, but our point is more about seeing the expression, and at this point we just believe that if we find the peak of the BFP fluorescence, then it is the peak of our production.
For example, do they have equivalently strong RBSs, or did you think about the maturation time for the BFP, for example?
Yes, we use the same kind of RBS for the same expression strength, and I think that's about it. Yeah, thank you for your question.
Is it possible to get P-band synthetically, not from E. coli, like as a standard, and then you can compare how much you're making?
Do you mean how the concentration we can create with?
P-band's a short peptide, right?
Yes.
A few amino acids. Is it sold commercially by anyone so that you can compare how a synthetic peptide bought from the company compares to the lysate that you're producing?
It is because P-band is a newly found substance, so we didn't really find any commercial synthetic way to synthesize P-band. So yeah.
All right. So
there
was no possibility of a standard. So
not that we know of.
Yeah. And then as a kind of second question was, you said in the presentation that it works and it is specific, so that only the species that the P-band is intended for are the ones affected. But
yes.
What data do you have to back that up?
We did testing. We did field testing in like the farms we cooperated, and we saw that with the blue light attraction, there would be many different kinds of moths. But after a period of time, the more powerful attraction would be the same kind of moth, the species of moth we're intended to attract.
Is that data on your wiki about the numbers of the different species that have managed to go because of your specific P-band?
Well, no, we don't have the exact data with us right now due to the time limitation, and also because our target insects have, at this season, they're already they're less active. So by the time we created our entire system, we didn't have time to complete a very, very detailed field testing to create actual numbers and quantify results, but we did observe the phenomenon. Yeah. Thank you.
With your device, like, were you suggesting that, like, everyone should have one, so like we all have these devices in our backyard, like attracting, well, wherever the moths are, So if you have like hundreds, like maybe like thousands of people with these devices in their backyard, and then they're attracting all these moths, you'll probably have a, I mean, you are aiming for a depletion of moths, right? But what are the consequences of that? Like, did you think about maybe like ecological consequences? Like, could you kill too many moths?
Yeah, we have thought about this question for a long time, and because our system is intended for smaller range attraction. So we would try, if this is commercialized and everyone is using it, we would probably launch some kind of regulation or some kind of standards to evaluate if your environment is suitable for our system, so that maybe like in this area there are 4 houses and they are all bothered by moths, but one of them is already using our system, then maybe we would tell them that, oh, you wouldn't need our device because someone else is already like clearing off all of the damage. So you can avoid this kind of problem by putting more thorough evaluation for each of the purchases and like how you would. Yeah.
Just a quick one. First, very entertaining presentation. Thank you.
Thank you.
And just in the video you showed, you had covered the device. Can you just explain why you need to cover the device?
That's a good question. You have a very sharp eye. Yeah, we covered the device with aluminum foil so that the light will only come out from the duct, the opening in the bottom. So the moth wouldn't like crash to the wall because it's giving off blue light. It would just go into the opening from the bottom. Does that answer your question? Yeah. Yeah, exactly. Thank you.
Okay. Thanks for your nice presentations.
Thank you.
Yeah, my question is, your device can kill some insects that may be harmful to the agriculture or to the human beings.
Yes.
But have you do any experiment to show your device will probably it can also kill some other insects that is not harmful to the human beings, like butterflies and bees? So maybe possible. So have you do any experiment to verify or testing your device that it only can kill some insects is harmful and will not kill some other insects is not harmful? So maybe you need to do something about this.
Yeah, this is a very good question, and we did think about this, as I mentioned. Because P-band is specific, so when we have the very powerful attraction of pheromones is already species specific, so we can actually separate our attraction into 2 stages. First is when we attract the first insect that's going to help us using the blue light, and that's not specific, but it's a smaller range, so it would affect much fewer kinds of species. But after the P-band reacts with the insect, then it's specific, so we don't have to worry about that phase. But for the blue light attraction, we would do evaluation in the surrounding environment, and for now, all of our target insects are more nocturnal, so they are more active at night, and they will be attracted by blue light. So we would make sure that there wouldn't be other non-targeted insects that would be